Okay. So good morning. Well, for us, it's good morning. It's okay. Good morning, everybody. So thanks for coming by for our talk about lightning fast genomics using Spark and Hack. I hope that most of you have seen the Spark introduction and so on, because we are going to be very fast on this track. Maybe we'll focus on the genomic and the Hack part. So, a few words about ourselves. So, I'm Andy, on the left side. Most known as Nutsap on Twitter. My company is NextLab. I'm a judge organizer. I'm also organizing DevOps for Kids, which are events dedicated to kids. I'm a mathematician. I did some computer scientist studies, and I'm learning data for several years.
I'm Xavier. I have a background mostly in academia for years, doing physics, then moving towards data analysis, genomics analysis, and lots of data, so distributed systems, and we'll see where it goes after that. So today we're gonna talk about a domain application that is genomics, something that we like. I don't know if there are any bioinformaticians in the audience. No? Yeah, 1, 2, 3, 4. Okay. So the idea is that we start with a kind of reminder about what genomics is about. I think, from my point of view, the most relevant application of genomics, of course, is healthcare. Genomics applications deal with medical diagnostics, drug responses, which means that from your genomes it's possible to target specific drugs that you will be a better responder to, and understand diseases mechanisms, which is also very important because genetic diseases usually are not really understood and they are chronic and really painful. When we think about genomes in terms of more data stuff, the human genome is made up of actually 3 million long sequences of what I call bases, which are small molecules. And in the human population, one of each 1,000 bases is actually variable. And those variations makes what we are each in this human population. The purpose of genomes in biology is actually to help encode biomolecules. And those molecules are really in large numbers and make a kind of, if I can say, biochemical soup that every living entity is. And it's a very complex entity in itself because they interact with each other and with the environment, right? So biological systems are very complex. It's a lot of data, a lot of interactions. So there's a lot of work to do to understand how it works. In terms of state-of-the-art, the sequencing technologies really emerged in the 70s, and since then the progress has been really incredible. And this growing capacity is actually not, I would say, caught up by the Moore law, which means that in terms of IT, we may have a problem there, right? It costs less and less. The price of sequencing a genome drops very, very fast, and there are more and more data that are being fed into the databases. So today what happens is that IT is becoming a bottleneck in terms of cost and latency. And you see here actually the cost and effort in yellow required in terms of IT when you want to process genomic data. It's growing, so we have to do something. And this thing shouldn't be sacrificing the data we collect. We don't want to cut off, we don't want to sample the data. We collect more data, we use it, right? Now the problem is that since all those bioinformatics efforts started in the 70s, at the time it was not really designed with scalability in mind, right? And we relying on a stack that is traditional, you want to look at what the file is, so you do a cat to grep, so on. And it's not really adapted to really massive amounts of data. And the traditional edge performance computing approach is not a fit. It's a data-intensive application. We want to move the program to the data, not the data to a processing cluster. Okay. And the future of genomics means that we will sequence more and more. Every individual might be sequenced in a, well, maybe remote or near-term future. But new sequencing technologies are arising, dropping the cost even, the time it takes to get the data, and we might sequence stuff as needed. You have a sickness, maybe not really bad one, we sequence to see what happens in your digestive system. There are many bacteria, and we can get information about them just running sequencing reactions, right? But you have to process that. So in terms of a medical condition is what? It's an interaction between your genome, which is pretty static data, and your environment, what you did in your life. So it's a very complex system again, a lot of things to do. So genomics is a need of scalability. And that's why Scala and Spark are providing tools to improve the situation. And it also requires simplicity and clarity, and that's where the efforts like AMPLab provide solution as well.
Yeah. So yeah, that's the background of genomics. As Xavier just said, we need some clarity in the genomics work. And why it's not clear actually for now, because the files are just regular text files. Okay, so it's quite hard to have something quite homogeneous, shared around, and that can be completely interoperable between the systems. So what ADAM provides is a API that is clean, and to do that, they refactor all the data are shared. Yeah, this way? Yes. Better? Okay. So yeah, the problem is the text file, so they provide a way to access the data more efficiently, and to provide efficient access to the data, they need to do it in a distributed manner, right? And text files are not easy to distribute, because since the sequence is very long, it's 3 billion items, so it's very hard to see where the cutoff must be done. So they introduced Parquet, and for that I will give an introduction to Parquet. So it's a columnar storage. So we have a matrix of data. So each row is a genome, and we have two options to represent it into a hard disk. You have the row-oriented fashion, that is just picking all the columns and appending to the text file or the binary file, and the column-oriented, which takes each column, append it, and then switch to the order. But actually, Parquet is a columnar storage, so it takes advantage of three things. By choosing the columnar storage, is having the data that are homogeneous, okay, but they are also collocated, right? Because you have A1, A2, A3, etc. That means that you have ints, for instance, that are collocated. So you know, if you know how many ints there are, you can switch from A1 to B1 very efficiently. But if you want to have access to all the rows, but only picking A1, that means that the column A, we just have to range access the first part of the disk at once. So you will have the first column. So select A from table B, for instance, or T, let's say. It's very efficient. And since you know the types that are collocated, you have a better encoding. Because you know, okay, this is ints for 1,000 values. So you don't have to repeat the types, for instance. Okay. Now, how could you define a Parquet schema? Because Parquet is made for nested structures, as genomics are actually. Xavier will give more insights into this. But actually, Parquet can give a very neat presentation of nested structures. Like here, we have a document. It has a doc ID, which is required. It has an optional group of links. Links is another type. And the links can have two repeated values backward and forward, and the things going on with web language, string code, etc., etc. So you can repeat, and you can group, and you can have a optional value. Actually, it's very simple, and we can see it as a Scala, case class in some sense. So Parquet is very efficient in structures. So it represents the nested structures as a tree. Empty levels are actually nothing more than just pruning a branch. So you have 1,000 documents, and they have optional values. So rather than, in JSON we have this empty bracket to say that actually there is nothing in there. So in Parquet they will try to just register that there is an array there, but nothing is embedded in there. Repetition is just metadata, and since we have types, we can have a very safe and fast codec. Like, I don't know, the JSON one in Play, or I can remember the other one. Anyway. Okay, now more in-depth information on the representation. So we have two objects there. By the way, this report, sorry about that. We have two objects with level 1, level 2, level 1, level 2, etc. A, B, C, D, E, E, F, G, H, I, J. And how you can represent it is just a simple row of data, right? You have 0 because it's an object. A is the first value of a level 1 in level 2. B is a value in the second level of in the structure. So we have level 1, level 2. So 2B, 2C, 1 new array of level 1, 2 E, etc., etc. So it's very efficient. It's very easy to parse, it's very easy to reconstruct, it's very easy to store. It's very hard because the quality is pretty poor. Okay, so we have the structure, we know how to represent it. Now, how is it stored? Why is it efficient, actually? It is efficient because you can split the file, let's say, whatever you want, almost. You can split by rows, but you can also split by columns. That means that you can have plenty of files distributed into HDFS, for instance, but the metadata for the file is stored at only one place on each partition, and for each column chunk you have one metadata there, one there, one there. That's it, and it's completely type safe. So yeah. So we can, let's say, take all the D nodes in HDFS and ask for the column A in the table T, and we take the column 1 in partition 1, the column 1 in partition 2, etc., etc. So it's quite fast. So this is for the schema, how it's stored in the disk. Now, how to represent it in a clean and easy fashion. We can just write an ideal. It's Avro. It uses Avro for the storage, for the encoding, and it will convert it into the JSON schema, which is always shipped in the protocol. That means that since the JSON schema is each communication with the client, any client can really easily create an interface on top of it. Then, as I say, this is just part of the protocol. It's in the serialization, okay? So we need less metadata in the structure because the schema is provided once, and then from there we can generate a lot of serializer and serializer. However, the idea just convert into a JSON representation, but actually the shipping is done in binary, so no text. No text is involved in there.
Okay, now it seems that we are going to store structured data in an efficient way and distributed way. The talk is about the use of ADAM, and the credits for the development of the start of the development of ADAM is actually the AMPLab at Berkeley, and they have a number of projects related to genomics, And now let's get back to another view of what really is a DNA sequence. You may remember that DNA sequence is a double helix thing, which is a molecule, actually a chain of small molecular elements, and there are 4 kinds of elements which will be labeled T, A, C, and G. So from the modeling point of view, it's just a string, right? Where characters are taken from a 4 letters alphabet. So it's pretty simple. The mess comes from the fact that when you sequence the genome, 3 billion characters is not that much, that's okay. But the reality is that it starts from a molecule, then the reaction that happens in the laboratory is that it's chopped into random spans of, let's say, between 100 and 200 bases. And all these are mixed into the tube, and they are sequenced, so there's a reaction that gives you actually the actual string corresponding to each of these fragments, but then you have to reorder them, right? Which means that if you know the reference sequence of the human genome, you have to find where they fit. So that's quite a number of realignment of these fragments. So that's quite a lot of work. But if you do that for those, let's say, 2 million reads, which are those fragments, you don't cover the entire genomes because they're taken as random, right? So you have to do that quite a number of times, and each position may have some poor quality, so you need to do that quite a lot of times. So that's really a lot of data to be reorganized. And the AMPLab project of the AMPLab project is actually doing that. So you'll see that in more detail because we use that. Then, what's interesting in genetics is the variations. So you have to be able to identify them, and that's the job of another project, which is Avocado, but I don't enter the details here. Now, what happens when you want to represent data, genomic data? You have actually, you don't see the thing here, but there are committees that they meet and they say we have to organize the stuff. So they write specification for the formats that will be available for the research and the use of these data, and they produce a document which actually describes what will be stored and how it will be stored in a text file, right? And there are such standards, SAM, BAM, VCF. There are strategies to index those files, so that's not that bad, but you rely on people developing libraries in order to be able to use that specification. So several teams are doing the job, that's okay, but you may have some instability because some libraries will not support all features, so it might be a bit hacky to get the job done. In terms of distribution, there are efforts like Hadoop adaptation of the specification. They scale to a certain extent, but that's not perfect. What AMPLab is doing is that it's actually used a schema-based approach, right? So they define these records and the relationship between the different entities, let's say, and libraries are generated for any language. So there is no doubt that as long as the schema is good, any language will support the specification. There is no storage specification here. You can store in any structure you want. Relational database, Parquet, whatever. And so, you got that actually the system uses Parquet because of its features that Andy just exposed before. Now, what is the AMPLab API? AMPLab actually provides the AMPLab context, which gives you functions basically to read the data on the HDFS. And hopefully you went to the Spark introduction because I will not explain what the Spark context is, except that it's the object that gives you access to your cluster or distributed environment. But you see that thanks to the design, you actually can use the Spark context directly to call your Adam context functions. So the environment is very easy to use for the Spark users. It also provides, of course, the Scala classes that are generated from the Avro schema. Data is loaded as a resilient distributed datasets, and you get access to functions on these RDDs that is writing to HDFS using these genomics formats and functions to manipulate, specific to manipulate the genomic data. For example, reading a data set that is saved in the ADAM format from the HDFS is as simple as the Spark context, ADAM load, and the URL of the file. Okay. Now they at the AMPLab they've done some benchmark, and they could show that actually it scales pretty well with the number of servers that you activate, while the traditional approach, even on the Hadoop framework, is actually saturating at a certain point. The reason being that the data is not so well distributed among the cluster servers. And, of course, the code is simpler, because you're writing in a functional way. This makes the code more compact. Right.
Cool. So, now we know more or less what is ADAM. So, with Xavier, we thought, okay, so what could we do with this distributed environment? Okay. And there is a project, 1,000 genomes, and I'm going to give some insight into this project because it's very cool when you are doing genomics data and genomes-oriented algorithms. So at first we wanted to have some data. So since genomes is very related to your private data, it's very related to your health, it's still thought as a private data. So we had to finally find some data, and of course there are options like the 1,000 genomes. And the 1,000 genomes actually is targeting to sequence completely 1,000 people. And they reached this level 5 years ago, so, or 3 years, I don't remember. But now they're retargeting their number to 2,000, because 1,000 is cool, but more is better. And what they are doing this sequencing, completely anonymous, and they are recording from where the guy came. So we can have a mapping between the origin and the type of sequence that we can have. So this is the browser that they are offering us. So it's completely awful, and actually you can slide, choose your chromosome or whatever, and then you have to go to FTP and download the file. Okay. So that's the legacy stacks that we were talking about. Okay. By chance only, the NCBI in the United States thought, okay, this is crappy. Let's take all the data and put them into S3 as a public dataset. So at least we can download the data from S3 now, which is kind of cool because it's really easy to download from S3 and put it into an SE2 instance that contains as a pre-built HDFS installation. So actually, how many data? 200 terabytes. For 1,000 guys, 200 terabytes. That's why, because it's not only the sequence, but all the information that we collected while sequencing the guy. The 30 sequences, so it's 3 billion times 60, for instance. So it's 90 billion information, but we have to know, okay, which precision did we have on this variant, and so on and so on and so on. So it's huge. So the goal of this project, as I said there, is that they wanted to see the genetic variation between the population in the world. And so we took this dataset, publicly available, and they were high quality, and we tried to validate ADAM itself for several projects that we are involved in. And the question that we tried to answer is it possible to predict the appartenance of a genome to a subpopulation in the world. So it's just a toy, right? There is nothing more than just trying to do something with data, just to prove that data is kind of cool. And to do this, we try to apply unsupervised algorithms, which are generally more intensive in resource consumption than the others. So with this data, we can run an unsupervised algorithms on the massive number of genomes that we had, right? So the idea to find the correspondence between the genome and its population is trying to find the clusters and trying to see from where they may came. And why it could be interesting after implementing the toy, we say maybe it has a added value, but we thought that maybe it can be interesting to see what is the gene flow, selection, and so on, or how the gene is evolving in the different populations in the world with the different mixing that we are seeing now. Okay, so 200 terabytes, as I said, and in order to, how do you say, to not spend too much data in EC2, we try to take a subsample of the 200 terabytes, which is only the chromosome 6, the 6th chromosome. And how to do that is very simply, we install S3 CMP for who knows, and we download the file from S3. And that's more or less it. And the chromosome 6, as you can see there, is worth of 170 million base, which is not so small.
What is the data we actually look at? We look at genotypes, and a genotype is actually something that points to a variant, and the variant is telling you that there's a position on a chromosome that is variable in the population, okay? And it tells you also which are the alternatives, okay? And most of the time you have two alternatives because mutation occur at random, and in order to have mutation at the same position, it's very unlikely. And we selected those positions with only two possibilities because it's simpler to process them. Then a genotype also points to a sample, someone that was selected in the population. And this person, I remind you, has 2 copies of each chromosome. Let's forget the sexual ones. And so, when you have a sample and a variant, you look at what is the state of the variant on each copies, and you get actually 2 values that are called the alleles of the genotype. What data we have also, of course, is a mapping between the sample ID and the population in a file. The project has maybe 30 population, I don't remember exactly. We selected 3 of them to take a subsample just to check how the technology works with the data. So the data structure. As Andy said, we start from the 1000 genome data, which is obviously stored in the specification format, which is VCF. It's a text file where you have many lines. Each line contains a number of genotypes. And it's converted into AMPLab genotypes with this instruction. So basically it's loading the VCF file, and we transform it into a collection of genotypes. So we have obviously to flat map the stuff and convert it to the data type, genotype in ADAM. Then we save it. It's distributed on our cluster, and we're ready to work with the data.
Yeah. And we switch from a 8 gigabytes file to a 22, I think.
So uncompressed, the VCF file is 70 gigabytes. Compressed, it was 7, 8, and we get a compression factor with the Parquet format. So it's also efficient in terms of compression.
How long does it take to convert a VCF file or BAM file into ADAM?
That's the hard one, because the band of SAM file are not distributed by essence. So this task can take time. Okay,
But we're talking about maybe 1 hour.
So it is faster to convert VCF, that's why we use VCF and not index back.
We read what's painful is actually to decompress the VCF first. There's 20 minutes waiting, taking coffee probably. Then we have to load it into the AMPLab, which is also painful, it's not yet distributed. But then it's self-distributed. So from that point, we are just distributed and efficient. If it's 4 nodes, 40 nodes, or 400, you see, that's why we have to take this step. We have to distribute the data anyway. It's not produced in a distributed way. So as I said, as Andy said, we will make some clustering of it. Very naive, admit. So we decided that we would use AMPLab, of course, on top of Spark, and clustering algorithm is K-means. K-means is an algorithm that actually starts with random yes. You select the number of clustering ones. Random items are selected. There are the centroids actually of the clusters, and the clusters are made by selecting the samples that are closest to the centroids. And then you recompute the centroid, which is actually the average position of the cluster, and you iterate over and over until you have convergence. In our case, we work with, as I said, genotypes, which are actually exist in 3 states, because you have 2 letters, maybe they're the same of one kind, maybe they're the same of the other kind, or they're different. We cannot make the distinction between the 2 chromosomes, so we have 3 states, it's encoded as 1, 2, 0, 1, 2, and we have a number of variants. Okay? In principle, you should use Manhattan distance, which is the edit distance, how you go from one combination of letter to the other. But because the algorithm is relying at lower level on the Breeze library and L1 was not implemented, we said, okay, we'll add that later. So, how does it work to get K-means running in Spark? It's pretty simple. You've got actually access to machine learning algorithms in AMPLab. That's the K-means here. The output is a K-means model, and you have a structure to organize your data, let's say, which are actually vectors that are relying actually on the Breeze vectors, the Breeze library. And it's just, K-means train data set, the number of clusters and the number of iterations. How do you organize your data to get this thing working? We saw that there was a data frame. So we start here from a long connection of genotypes. So it's basically a sequence, and we want to make it a table with each row being a sample and each column being a genotype. Okay? So we simply have to make a group I, where the group is the sample ID, and then we have to store as value an array which is the different alleles. Okay, and we have to be careful, of course, to sort it every time in the same order, just for this to make sense. Okay. Then the prediction. Once we have the model in our hands, the prediction consists in calling the predict on a vector, which is actually the features value of a sample. And so we can see that for each sample we assign a cluster number, and we'll try to see if it maps to a given population. The results are so we have 3 clusters. We see that every British sample aggregates into cluster 2, so we can guess that cluster 2 is British samples. Here you have American with African ancestry, and they cluster pretty well in the group 1. You have a number that are mislabeled, and people from China are all segregated in the same cluster. So it works pretty well. The data set we use is not the whole genome. We took a subsample because we had no time to run the bigger stuff. But you see that the system is scaling, so we'll be able to go further.
Yeah. Okay, so how we ran that? We ran it on EC2, so it was probably a normal army that just for time spend, a whole bunch of money that we may have to spend. Anyway. Okay, so we started with only 3 instances. M3X large, which are worth a 4 core each and 15 gigs for each node. That means that we had the power of 16 cores and 60 gigs and our So the performance. So actually, this is mainly the different stages that Spark will launch in the cluster. Okay, and as you may see, most of them are around less than 5 seconds, and there are 1 or 2 that are around 2 minutes and 4 minutes there. Okay, 4. But at least the whole trick takes around 10 minutes to run.
How many genomes were there? I mean, from how many genomes did you analyze? From all 1,000?
No, we took 3 population. That's 250 genomes.
So it's a sample of 250, right? Genomes.
Yes.
And
we took a slice of the chromosome 6 that was 1.5 million bases, resulting in 20,000 variants.
Yeah. That's for the 4, the customer size 4. Yeah. But we said, okay, maybe we can scale it up a bit for a little while, and we switched to a 40 cluster. In Spark it's really easy, so just changing the number in the Spark EC2 command. Okay, just waiting for 10 minutes, calling Amazon, okay, could you raise my limit to 40, please? And 5 minutes after, we had our 40 nodes available. And actually, so we switched to 160 cores and 60 gigabytes, 600 gigabytes of memory available for us. And it was amazing. We were all excited during this. And actually, the 4 minutes, almost 5 minutes task switched from this 5 minutes one to less than 1 minute. So we scaled by 10 and we reduced by 5. It's not linear, but at least if you want to scale up some algorithms, we can see that Adam enables it. And it's still young. Adam is very young. It's only in release 014, so it's very, very young. And the other one, which was around 1 minute, slows down to reduce down to 2 seconds. So we can see, scaling by 10, we have a factor of performance around 5, let's say. Okay. So some conclusions on this work. So mainly the idea was to validate ADAM rather than all the legacy stacks that we can have and use. And at least what we saw is that ADAM provides tools to manipulate genomics data very efficiently and easily. I mean, the code that we shown was 40 lines of code to read the data, convert it, save it to HDFS, load it from HDFS, applying k-means, prediction, and draw a table. Man. Okay, in Perl, C++, in MPI environment on HPC, it would take years. Okay, so, and this is mainly because of the beauty of ScalaIO, this is why we are here, and the machine learning library, which is evolving quite fast in a good manner, if you like.
So of course, what we did is pretty naive. It's not genomic research. It's more technological evaluation, right? But it tells us that we should go to process the whole stuff because it seems really to work. Give this to research teams and to apply really state-of-the-art algorithms on the genomic data, and of course cross this data with something else than just the origin of the sample population to do something that is relevant in terms of healthcare. We want to thank ScalaIO for giving us the opportunity to talk about this venture, and at AMPLab, Matt and Frank, and also Vincent Botta, who helped us design the work here.
He gave us this map animation. So that's all. Okay. Nice.
Thanks, guys. Any question?
Okay. Well, actually, what I see in a Scala world that there is a lack of biological libraries in Scala. So, which library ADAM relies on in terms of representing sequencing annotations? Does it rely on some BioJava, or it implements classes on its own?
It's on its own, because actually they say we have to start over with a schema, because the problem is that we have to distribute data, and to do that we need a schema. And so they had to write the schema first. What is the data we want to put it in? What are the relationships between the entities we're dealing with? And then we generate the libraries with the other tools, actually.
Okay, and I also spotted such library called Spark sequence, Parsec. Will it be integrated with ADAM? Or you don't know?
I don't know. I've seen that a couple of days ago, you're right. So it's worth looking at the efforts, and I'm sure we'll come back to AMPLab and talk to them about that, sure.
Okay, and the last question, how much in terms of money did it cost you to run the last example on that huge cluster? Approximately, because...
40 euros?
40 euros?
Yeah. Sorry?
No. So we prepared the code, obviously, with that one. But on the night we decided we would run the stuff on the large cluster. It was up for 2 hours.
Yeah.
And we had to solve some issues. I passed the very detailed DevOps issues. It should have run in less than in 1 hour. We're talking about $20.
Yeah.
Thank you.
Hey guys, what are you using to do the conversion from Avro to Scala classes? So what are you using to do the conversion from the Avro schemas to the Scala classes? Because I know there's like you can convert into Java classes, but there's no nice, as far as I know,
For
way.
the Avro part, well, actually the code is generated as a Scala already, so there is no Java involved in this.
But what's, are you using like a library or something to basically read the
Actually,
do you know which one?
I guess it's Scrooge, but
You're using Scrooge.
I cannot swear it because obviously it's in the build system, so we don't see it actually.
Yeah, Scrooge is for the first, but it's something like it's just worth launching the command, put it into the SBT builder. You don't see it.
Thank you.
As we would like.
Yeah, Scrooge is mostly used for this kind of stuff.
Yeah.
Any other question? No.
So I saw you use MLD. How is your feeling about this? Because my feeling it is really in infancy. You can't do many things. It is very basic. For example, for chemistry, you can't set some properties. It's just like that.
Yeah.
And how is your feeling?
Indeed, but it was a toy, right? Okay, so it does the job, right? We know that it's not scikit-learn, or it's not R, or whatever. we know that it has some limitations, so that's why we provided the number of iterations that we wanted to run, and we didn't wait for the algorithm to convert directly. So yeah, we know that AMPLab is not, yeah, it's not providing the whole flexibility that we can have, but at least it does the job for us.
Do you have problem for saving the model, for example? You know, we usually train and then predict separately. How do you handle that?
We said it was a file HTML.
Yes, that's pretty much.
So you just realize the model to HDFS and then you load it.
Yeah, as an object.
And no problem with version changing.
It's a toy, so no, it's not. It's only a product. So we wanted to prove that the technology is good, but we didn't went there. It's not working.
Just one question for me. Did you want me to do a standalone Spark cluster or on your own, for example?
A standalone what?
Your cluster, actually, Spark cluster on EC2, was it a standalone cluster?
It was, it was. Using the Spark EC2 one. No measures involved there.
Okay. Any other question? Good. Well, thank you, guys.